So welcome back to Primate Pro, and we'll continue with the lesson Cell Unit of Life. This is the lecture number 6, and I think you all have enjoyed a Christmas vacation and hope all are doing well. So we'll start from the endoplasmic reticulum today, and we'll continue with the Golgi bodies. So let's start with lecture number 6. So let us start with endoplasmic reticulum. Endoplasmic reticulum. Okay, so endoplasmic reticulum, it can be segregated into cisternae, vesicle and tubule. So endoplasmic reticulum can be differentiated in cisternae, vesicle and tubule. So what is the function of endoplasmic reticulum, where it lies? Basically the endoplasmic reticulum lies inside the cytoplasm. Okay, inside the cytoplasm it is a network of membrane of cell. It is a network of membrane of cell. Okay, so it actually locates inside the cytoplasm. It is a network of membrane. How it looks, how the endoplasmic reticulum generally looks. Okay, I am drawing a picture of it. Okay, this is the endoplasmic reticulum. I am drawing the picture. Look into the picture. I will describe it here. Okay, this is the inner membrane. This is the inner membrane. So this is the outer membrane. This is the outer membrane. Let me do this less 1.5. Okay, this is the inner membrane, this is the outer membrane. Okay, now here this is the rough endoplasmic reticulum. This is rough endoplasmic reticulum. This is the RER, that is the rough endoplasmic reticulum. Rough endoplasmic reticulum. Okay, why it is named as the rough endoplasmic reticulum? The rough endoplasmic reticulum, it is named because the ribosomes are generally accustomed here. The ribosomes are situated in the endoplasmic reticulum, and the function of the ribosome is to make the protein. It will synthesize the protein, because the endoplasmic reticulum, those contains the ribosome in it, it converts into rough endoplasmic reticulum, and those endoplasmic reticulum do not have any ribosome. Those are the smooth endoplasmic reticulum, and they are generally functions as to produce lipids. Okay, this is the rough endoplasmic reticulum generally contains cisternae. Okay, here the luminal spaces, luminal spaces inside ER. Okay, here extraluminal spaces This is the cytoplasm. Cytoplasm is here. Okay, the smooth endoplasmic reticulum. The smooth endoplasmic reticulum or the smooth ER, vesicle and tubule, vesicle and tubule. Ribosome is absent. Ribosome is absent. Ribosome absent. Ribosome absent. Okay. So in smooth endoplasmic reticulum, ribosome is absent. Their functions as smooth endoplasmic reticulum functions to produce lipid. Okay, now if I say to give a difference between the RER and the ACR, the RER and the ACR. Okay, so the RER and the ACR. RER means RER and the ACR. That is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Smooth, and this one is rough. So they contains ribosome. The RER contains ribosome. Ribosome present. Here ribosome absent. Okay. Here it is continuous with other nuclear membrane. It is continuous with outer nuclear membrane. Discontinuous with outer nuclear membrane. ये आउटर न्यूक्लियर मेम्ब्रेन के साथ कंटिन्यू नहीं करता है. ओके, they functions in the synthesis of protein. They function in the synthesis of protein. So RER functions in protein synthesis. Lipid synthesis. Lipid is being synthesized by or synthesized in the ACR. Okay. So number 4, secretion. Secretion. Protein formed by RER released outside the cell. Protein is formed by RER and is released outside the cell. Okay. Glycogen synthesis. Okay, glycogen synthesis. Now the intracellular spaces inside the cell. Give it the heading, the intracellular spaces. Intracellular spaces. This is inside the cell. Inside the cell. Okay. They can be of luminal and extraluminal. Okay. So electron microscope study. What does the electron microscope study? When we study the the intracellular spaces throughout the electron microscope, we can differentiate into two categories. There are some luminal spaces and there are some extraluminal spaces. इलेक्ट्रॉन माइक्रोस्कोप के द्वारा हम जब देखते हैं इसको अंदर से, तो ये इसके साथ ये लुमिनल स्पेसेस कुछ दिखाता है और कुछ एक्स्ट्रा लुमिनल स्पेसेस भी दिख जाता है. तो ये इलेक्ट्रॉन माइक्रोस्कोप के साथ इलेक्ट्रॉन माइक्रोस्कोप स्टडी नेटवर्क reticulum of tiny tubular structure. टाइनी ट्यूबुलर स्ट्रक्चर के एक नेटवर्क रेटिकुलम दिखाता है. कौन सा? ये ER. और जो नेटवर्क और रेटिकुलम है एक्सटेंडेड है, बहुत ज़्यादा नेटवर्क है, ज़्यादा से ज़्यादा एरिया तक ये फैला हुआ है. जब ये इलेक्ट्रॉन माइक्रोस्कोप के अंदर से दिख जाता है तब. So extensive, that is extended. Okay, और ये है tiny, small. तो ठीक है ना. उसके बाद हम आते हैं गोल्गी बॉडीज. ज्यादा डेप्थ में जाने का कोई जरूरत नहीं है, जितना आएगा उतना ही सफिशिएंट है. ठीक है. गोल्गी बॉडी. Who discovered Golgi body? ये पहले जमाने में आता था. कैमलियो गोल्गी. discovered by कैमलियो गोल्गी. ओके. सो ये गोल्गी बॉडी क्या है? ये एक बॉडी है जिसने कैमलियो गोल्गी बनाया. ये लिखकर कोई फायदा नहीं मिलेगा. ठीक है ना. तो इसको मैं पहले ड्रॉ करता हूं. ये जो गोल्गी बॉडी दिखा था ना, ये इसका मैच्योरेशन करता है. जो प्रोटीन बनता है ना, ये प्रोटीन बनाता है. लेकिन क्या है, जो इनएक्टिव प्रोटीन है ना, इमैच्योर प्रोटीन, ये राइबोसोम बना देता है. और इसको इनएक्टिव से और इमैच्योर से मैच्योर बनाता है ये गोल्गी बॉडीज. सो गोल्गी बॉडीज का फंक्शन है क्या? टू ट्रांसफॉर्म द इनएक्टिव और द इमैच्योर प्रोटीन इनटू मैच्योर और एक्टिव प्रोटीन. Okay, now this one is RER, that is the rough endoplasmic reticulum. Okay, now one is okay. This need to be a black ink. Okay, now I'm drawing another thing. What is this? Okay, these here ribosomes are absent. Here see, ribosome absent, cisternae. These are cisternae. Ribosome absent, cisternae. Here ribosomes are absent. Okay, so now transphase oblique maturing phase. Okay, and this is the concave phase. Transphase, maturing phase और the concave phase सब एक ही है. transphase, maturing phase, concave phase, सब एक ही है, ठीक है। तो ये नीचे क्या, टूवर्ड्स सेल मेम्ब्रेन, These are the Golgi bodies. Okay. Now these are this phase is the cis phase. ये cis phase है, cis phase और forming phase. ये कॉन्वेक्स साइड है टूवर्ड्स ER oblique nucleus. So what is cisternae? जो सिस्टर्नी है ना, ये कहाँ रहता है, ये दिखने में कैसा है? सिस्टर्नी आर द फ्लैट डिस्क, these are flat disc like sac like arranged concentrically near nucleus, discovered by Camillo Golgi कौन से साल पर? 1898. Densely stained near nucleus. These are densely stained structure near nucleus. Number of cisternae vary in different Golgi body. Okay. Now I need to draw another structure. These are the RER. Okay. Transport vesicle. Here RER, these are also transport vesicle. ट्रांसपोर्ट वेसिकल से ये प्रोटीन, दिस इज द सिस फेस, सिस फेस. हाउ द प्रोटीन मैच्योर्स? Now this lipid and the protein, L and P, the lipid and the protein, this lipid and the protein forms cisternae. This lipid and the protein forms cisternae. lipid and the protein forms, these are the transphase Golgi bodies. Okay, coated vesicle, cisternae, the RER and SER release transport vesicle which contain protein and lipid, which contain protein and lipid. 2. Transport vesicle carry P and L, that is the protein and the lipid moves towards cis region of the Golgi body. Cis region of the Golgi body moves towards the cis region of the Golgi body. Cis region of the Golgi body. Okay. Now the protein and the lipid, protein and lipid enters into coated vesicle of Golgi body from transport vesicle. Packaging material, these are the packaging material. Okay, now this is the cis side. Just remember, this is the cis side. Note, here give a note. Golgi body receive the material from endoplasmic reticulum, at which side? If this is the question comes, this is the cis side. So Golgi body lies in close contact, एकदम चिपक के रहता है. ये एंडोप्लाज्मिक रेटिकुलम है तो ये गोल्गी बॉडी है, ये चिपक के रहता है. Lies close association with endoplasmic reticulum. Okay, एंडोप्लाज्मिक रेटिकुलम से यह चिपक के रहता है. नाउ अगेन ये 4 नंबर जो पॉइंट है, these are 3 points. Point number 4, protein and lipid moves from coated vesicle to cisternae. Okay, protein plus sugar, glycoprotein, lipid plus sugar, glycolipid, glycosylation oblique glycosidation, modification of material, सिस्टर्नी में ये मटेरियल का मॉडिफिकेशन होता है, तो सिस्टर्नी में ये सब मटेरियल का मॉडिफिकेशन भी हो जाता है. तो point number 5 is protein and the lipid release at the transphase side of the Golgi body. Protein and lipid released at trans side of Golgi body. सो सीस में ये लेता है, मैच्योरिटी करके ट्रांस से ये फेंक देता है. ये अगर सीस फेस है, ये अगर सीस फेस है तो ये सीस फेस से लेता है और ये ट्रांस फेस से फेंक देता है. ठीक है. सम प्रोटीन रिलीज आउटसाइड द सेल कॉल्ड द सिक्रेटरी प्रोटीन. सो कुछ प्रोटीन है वो सेल के बाहर ही सिक्रेट होता है. ये प्रोटीन को हम लोग सिक्रेटरी प्रोटीन कहता है. सम प्रोटीन रिलीज्ड outside the cell called secretory protein, that is called the secretion. Now note, बहुत इंपॉर्टेंट है, ये याद में रहना चाहिए. नोट क्या है? प्रोटीन lipid. So here released outside cell, secretory protein moves to cell membrane present inside cytoplasm. Secretory vesicle from lysosome moves to cell membrane. The lipid moves to cell membrane. I will teach this more details in the human physiology. There you can feel the medical science. Intracellular target. Inside cell. Okay. Now liposome. Here note 1. RER provides protein for synthesis of liposome. Lysosome. Note 2. Lysosome formed from Golgi body. lysosome formed from Golgi body. Okay. So now you can say that there are two phases. There are two phases, phase cis and trans. So this is the receiving phase. Receiving phase. These are different but interconnected. And this is the delivering phase. Delivering phase. Okay. Now we come to lysosome. Formed by Golgi body due to packaging. Digestive enzymes in bracket proteins. Basically the digestive enzymes are proteins. Digestive enzyme helps in food digestion. Okay. This is a single membrane. Okay. So digestive enzyme protein carbohydrates, protease and lipases. Okay. These three are called the three trio. It is called as hydrolytic enzymes. They need water to perform or to function it, because these bonds are broken by reaction of water. So these are called the hydrolysis enzyme. Needs water to function. Okay. Work in acidic medium. pH of 5. All enzymes are hydrolyzed enzyme. All enzymes are in cell acid hydrolysis. Digestion of these are required carbohydrates, protein, lipid, nucleic acid. Okay, so I have continued till the lysosome, and make one thing note: lysosomes are called suicidal bag. It dissolves themselves also. So these are called the suicidal bag. Okay, so I have continued till the lysosome for today, and in the next lecture we'll continue from the later parts. In 2 or 3 lectures I'll try to complete this chapter, then I'll move to the next chapter and I'll continue with botany and physiology. Okay, I'll start with a chapter of physiology, and after that I will also continue with botany. So one botany, one physiology and one zoology. That will make the syllabus completed very fast. Okay, thank you. And if there is any problem in understanding the videos, do write a comment in the comment box. I'll try to make clear of that. Okay, thank you.